Okay, the next thing they always like to test for your this starch is the structural components of starch and why is that a good storage polysaccharide. Yeah? So again, we are always expected to know the structural feature as well as what is actually the purpose of it or enables it to function. So the role for it. Yeah, again, few structural features. Let's see. First structural feature we learned for starch. Your starch is actually very what, Katrina?
Large.
So large and what? Made up of many glucose monomers, right? How come must be large and made up of many glucose monomers? Yeah?
Because it acts as a storage.
No. If you think about it, if it's large, is it soluble or insoluble in water?
Insoluble.
That is the reason why. So the first one is large molecule and made up of many glucose. Purpose of this is that number one is insoluble in water. So it means that actually starch is stored in large amounts. Can be stored in large amounts. By cells that store it, okay? Without affecting water potential. Okay, how did I write this? Sorry, insoluble in water, without affecting water potential. Second feature, you must made up of many glucose for a reason. Why is that so, Rishika? You think about this. Do you think you can hydrolyze to release many glucose? Okay, so again, can be easily hydrolyzed to release many glucose. For what purpose? For what purpose? Yes, okay. So again, for respiration to produce ATP. Alright, so in other words, this enables what we call the large energy store. Next one, Sarah, what's another feature for starch that makes it a good storage polysaccharide?
OH groups are projected inwards, outwards, inwards.
Why? Why is that necessary?
Prevent cross-linking.
Why is that necessary?
To I don't know.
Think about this. You want to hydrolyze glucose, right? Cross-linking via hydrogen bonds, do you think that more energy needs to be overcome to break that hydrogen bonds? Is it easier to hydrolyze if you have more energy need to be absorbed? No one. You want to release glucose fast, right? Agree, for energy. So, purpose of this is why you want to prevent cross-linking for easier hydrolysis. Between chains for easier hydrolysis. Idea is for energy, yeah? You should know by now because it's still respiration. What else? We also have chains that is arranged in helices. Chains of glucose, when they are arranged in helices, same thing is for what? Compact packing. The compact packing is for starch, so it enables storage of large amount of energy in its small space. Okay? Last one. The last structural feature of starch is the amylopectin is branch. Think about this, whenever amylopectin is branch, right, you just need to hydrolyze this branch point. You straight away release short chains of glucose. So it means that it enables faster and easier hydrolysis. This is just a major thing. Okay, now next, I'm going to summarize now in a table, which is your triglycerides, okay? Because again, triglycerides and your phospholipids are the main things we always focus on. Okay, I'm going to do a summary on a table that separates structure, properties, function. Let's talk about all the structural features first. That differs in triglycerides and phospholipids. So triglycerides, hopefully you know, each triglyceride consists of what, Rishika? How many fatty acid chains? How many glycerol? So 3 fatty acids and 1 glycerol. Alright, so whereas this one consists of 2 fatty acids, 1 glycerol, and 1 negatively charged phosphate. Another structural difference between triglycerides and phospholipids is we learned that these phospholipids there is no other groups, whether carbohydrate or not, that will be attached to your glycerol besides your fatty acid. Oh, I forgot to talk about the bonds, sorry. Each one is actually consists of 3 ester bonds, my bad. Whereas your phospholipids consists of 2 ester bonds and 1 phosphoester bonds. Another difference I already mentioned, do not have other groups attached to glycerol besides fatty acids. This one, again, we already know may have other groups attached to phosphate group, maybe a nitrogen containing base, choline. Another one we should know, this is the properties. So properties, this is actually polar, non-polar, this one is actually charged. So let's talk about the properties now. One main property I already mentioned, this is strictly non-polar. Or you can say uncharged. This one, in fact, is amphipathic. How is spell amphipathic, Sarah?
A-M-P-P. What? A-F-P.
Then continue.
Amphipathic.
Yeah.
P-I-P-A-T-H-I-C.
Pathic.
No,
Correct. Eh? No, P-H-I. Spelling really matters. P-A-T-H-I-C. Okay? Means that you have both hydrophobic and hydrophilic regions. Understand? So again, we know the properties. This is strictly insoluble in water. This one, okay, the hydrophobic and hydrophilic regions are present. Last one I can talk about is the functions. Fats got many functions. One major function for triglyceride is obviously used as an energy storage. Later we'll discuss about the structural features. But now I'm just comparing between the two. So idea are, you can say, or respiratory, some of them can be used as respiratory substrate. Reason why it can be used as respiratory substrate, you should know it has very high energy per unit mass. Compared to carbohydrates. Other functions of triglycerides, I can say that it's used for, again, solubility of some fat-soluble vitamins. So it acts as a medium. That's why some of the substance can actually dissolve in only lipids. Okay? Again, you can say some of them can be a source of metabolic water, especially for desert animals like kangaroo rats. Or you can also say how it also provides aquatic mammals, okay, like whales, okay, with buoyancy. Okay. Another one I also accept is protection of delicate internal organs. That's why there's nobody with 0% fat. It's impossible. Okay? The famous one we always say is there is insulation, okay, to prevent excessive heat loss. can't think of anything already, but this is actually the main thing for your triglyceride. Phospholipids, you should know already. Structural component for membranes. So in other words, this is involved for what? Compartmentalization. An example for specialized conditions that I need to maintain is like the proton gradient between your inner membrane of your mitochondria and your matrix and your intermembrane space of your mitochondria. So this is your triglycerides, but we need to learn the structural features of triglycerides that makes it a good storage macromolecule. Okay, now for your triglycerides, there are just two structural features for you to take note of. First structural feature of triglycerides that makes it a good storage macromolecule is, I already mentioned down here, the high energy per unit mass. But the thing is, we need to understand what accounts for this high energy per unit mass, right? I went through this before, I don't know whether you recall. Rishika, do you recall what contributes to this high energy per unit mass? That means if I compare 1 gram of carbohydrate, 1 gram of fat, your fat always have, okay, more energy to release. So do you know what is it? Question. One thing, do you know how to recognize a fat molecule from a carbohydrate? I said that's a factor that we also learn in Set 3. Don't know? You? OH group. OH group in carbohydrate is about 1:2. But the OH group in your this triglycerides is significantly smaller ratio. So there's lesser bonds with O, right? If there's lesser bonds with O, isn't that more bonds with CH? So if I have more CH bonds, does it actually help contain more energy? So that one structural feature is that structural feature, it actually contains more CH bonds. When you contain more CH bonds than carbohydrates, like starch. Why is this actually important? Because this actually produces more ATP for respiration. And means that you actually have higher energy that can be stored per unit mass. This is exceptionally important for what? Animals that depend on energy for locomotion. Example, birds, you don't want to store them as glycogen, you rather store as triglycerides because they need buoyancy for flight. Or even in seeds, you need lighter seeds for dispersal, if it's especially by wind. So all this is necessary. Second structural feature we need to take note for triglycerides is it must be hydrophobic. What confers that hydrophobic nature? The hydrophobic fatty acid chains. The hydrophobic fatty acid chains, why is it important? Because you need your triglycerides to be insoluble in water. Why? So you can store in large amounts without affecting water potential of cells that store it. Please know the structure features of triglycerides. It's very useful. Now, when you're ready, I want to talk about this hemoglobin and collagen, because they are the other two types of proteins. But I'm going to summarize your collagen and hemoglobin in a form of structure and the properties and functions. So none of you have this. Let me know when I can erase. Katrina. Gerard, let me know when you're ready so I also can erase.
Distance.
Yep.
Can you shift the whiteboard to the right of me?
Right, huh? Sorry, this right, this right. Is it?
Oh,
This right?
wait, I screenshot.
You tell me your right or my right. Oh, you screenshot already. Oh, okay. You done? I erase class. I need to do proteins. So I'm just summarizing using this. Alright, now, like I said, I want to summarize. I'll start off with collagen first. So collagen, I want to discuss what is the structure and what is the property and the significance of those structure. First one, collagen. Type of protein. This one is again my structure. And then properties and functions. Okay, first one, structure. Structure of this collagen. Number one, you should know is a very large or have a very large molecular size. I also accept if you tell me, okay, large number of non-polar R groups, which is proline or proline and your hydroxyproline. So what is the significance of this property is, number one, if you're very large, means you're insoluble in water. This is necessary because you should know this collagen is actually a structural protein or fibrous protein. So what is the function for being this? The function, it means that it enables it to provide your structural support. In connective tissues, okay? Since that is where you mainly find collagen.
What is a large molecule mean that it is all
Okay, next one. I want to focus on the primary structure first. Because you know got primary, secondary, quaternary. So what is the primary structure? Primary structure of this consists of many tripeptide repeating tripeptide sequence. Of glycine XY. Please take note, X is usually proline, Y is usually hydroxyproline. What is the significance of this? When I have this, means glycine is in every third amino acid. Or we can say third residue. And it's small. Okay, so why is it important? So again, this is the property. So it enables it to fit into the crowded center of the triple helix. Triple helix is the same as triple collagen, huh? Okay? This allows it for tight packing of the triple helix. How come I also need hydroxyproline and proline? Proline and hydroxyproline are bulky, so it confers rigidity to the molecule as well. Because you should know, I will later form cross-links via covalent bonds. Okay, so again, this is important. Next one, quaternary structure. So what is the structure feature and how does it actually confer? Again, we have a triple collagen molecule. The triple collagen molecule consists of 3 polypeptides that bind tightly with each other to form the triple helix. That's why we have the quaternary structure. What's the property for this? Again, this one, it enables many interchain hydrogen bonds. This is within the triple helix. Which is the cross-linking via hydrogen bonding. Stabilizes the fiber structure of collagen. Okay, that's why it's able to be rigid. This is the property. But again, you also can tell me high tensile strength. Enables it to have high tensile strength. Both are acceptable. So in other words, the other property that we need to point out, since it's related, I have many of this interchain or cross-linking via hydrogen bonding within the triple helix, is because of why? This hydrogen bonds is between where? NH group of glycine as your CO group of another amino acid of another chain. Same. So what's the function then? This is the property, right? So function of this is that all this enables the collagen to resist the ability from snapping when you stretch your connective tissues. These two are property, function, okay? Another one based on quaternary structure, right at the top. So it's another kind of structure feature we can discuss. So for collagen, you have many tropocollagens, right? Imagine many tropocollagens, how do they bundle up to form fibrils? Is by covalent cross links. What property does it confer? This enables formation of collagen fibril. Which resembles to form collagen fibers. So, purpose, function, this gives additional tensile strength. This is for collagen. Huh? When you're ready, I need to do hemoglobin. Can I erase this part? Anna? Hemoglobin is a globular protein. So we need to learn what are the structural features of hemoglobin and then again how do they help with their function. First one, each hemoglobin you should know has 4 subunits, and each subunit has actually a heme prostatic group. The heme prostatic group contains a polyphenol ring that contains a iron 2+ ion. So this is the first one. Okay, subunit. Now, idea, what's the property? The Fe2+ is the one that binds to your oxygen. This is the property. So, what does this allow for function? Enables hemoglobin oxygen transporting function. Second structure, the quaternary structure. So in other words, therefore, it contains 4 heme groups. So in other words, they can carry 4 oxygen molecules. So increase the oxygen carrying capacity. Purpose of this, so it's efficient as an oxygen carrier. Again, under still the quaternary structure, each subunit you realize the hydrophobic amino acids, they are kind of buried into the interior, okay, and the hydrophilic are on the exterior. So this enables it to be soluble in the aqueous medium. It's still a structural feature. AA stands for amino acid. structure of hemoglobin in its quaternary structure is globular in shape. This actually matters because it allows many molecules to be packed compactly into the red blood cell. Please take note, not only is globular in shape, the 4 subunits are actually held together by non-covalent interactions. Now, you all recall there's cooperative binding, right? So that's why I need to emphasize, right? These 4 subunits, the alpha and beta, are not by covalent interactions because covalent bonds you learn are strong. Okay? So you need to have this non-covalent so you can actually change the conformational shape to allow cooperative binding. Non-covalent interactions are like minus ionic hydrogen. So, what's the significance of having these 4? Like I already said, allows for cooperative binding. There's the property.
So are the bonds between each subunit or within one subunit?
Between. Another one I can talk about is the heme group. Notice how, right, usually we talk about this, actually no, I talk about the subunit that possess what we call a hydrophobic cleft. The hydrophobic cleft will be served as the binding site for the heme group. We should talk about that instead. 5 is, we can say how each subunit is like a region, environment they create. Okay, so this actually provides the binding site for the heme group. To bind to oxygen, because you should know oxygen is non-polar. Okay, so all this will summarize between this proteins, hemoglobin, as well as collagen. Now when you're ready, I want to talk about the type of questions. Now what I did is a overall summary. Okay? For the main carbohydrates, they like to compare the main proteins and the main lipids. If they test you directly on all this type of carbohydrates, proteins, and lipids, very good, because you just vomit out. But you should know already, in paper 2, they also can test you application. That means they give you a new molecule, and then you're supposed to use and apply what you've learned to the new molecule. But do you think examiners will test you something that you do not know? No. Examiners will always test you anything that is based on what you have learned. They will never test you anything that is extra. Okay? So, one thing for this biological molecules, I want you to focus on question 1 first. I'm just using question 1 as a guideline. I'll let you read out this question 1 for your biological molecules worksheet that I just sent out. I'll give you about 5 minutes to just read and understand the questions before each one of you will help me. Notice how in this particular worksheet, I have attached enzymes as well, because enzyme is also a type of globular protein that is in your syllabus to know. Not just your hemoglobin, but also enzymes. So there are some enzyme questions that is also tagged in it, because sometimes enzymes may come together with biological molecules. Again, any questions I never get to go through, I will send you the answers, or the answers are actually tagged behind. Oh, Gerard, I just saw your WhatsApp. The insoluble in water can store in large amounts without affecting water potential in cells that store them. Do I need to repeat the one that you asked me?
Okay, thank you.
Okay. What I want to do well. Oh yeah.
Do question 1.
I just want to discuss. We'll verbally discuss. I'll let you read your question 1. You want to attempt? Go ahead, attempt. I'll go through another 4 minutes time. Let's go through, yeah? I know answers is already attached, but I want to discuss how we actually tackle these questions, yeah? Which is question 1. So question 1, right, they show you chitin is the second most abundant organic polymer after cellulose on Earth, found on walls of the fungi. We show you the they show you the chitin molecule. We learn nothing about chitin, even telling me there is presence of glucosamine subunits. But they did mention as similar to cellulose. So questions like this will show you, right? They will test you biological molecules. Sometimes it's another kind, but tell yourself it's still fundamentally based on what you will learn. So cellulose is what? Your linear chains, right? Cross-linkings via hydrogen bonding because each adjacent is rotated 180 degrees. Let's see, is there similarity? Do you see each adjacent subunit of glucosamine is rotated 180 degrees? Does it look like the, again, polar groups, the N, polar, they purposely write polar to let you know, N acetyl groups are projected all directions. Do you think can also have cross-linking to form bundling? So they even show you in figure 1.1 that it's formation of fibrils and eventually can assemble to form fibers. Right? So is it similar? Yes. So one thing we see here, with reference to figure 1.1, they asked me to name the covalent bond between and describe how this bond is formed. This is one typical question that just now I said. Whether is it your carbohydrates, your proteins, your fats, you always must have this. First of all, we should know the covalent bond is exactly the same as what you have for your cellulose. So covalent bond, again, quick one. Can you tell me, Katrina, is what kind? Very quick one.
Beta-1,4-glycosidic.
So it's beta-1,4-glycosidic bonds. But my question is, can you tell me, Katrina, beta-1,4-glycosidic bonds is always formed by what kind of reaction?
Condensate.
Yes. Whenever you write condensation, there is always a key phrase that you have to write for exam. Do you know condensation is always, what is that tag, that key phrase? Loss of
1H2O.
water molecule. You always have to write the name of the reaction and loss via what kind of molecule. So in the answer sheet, again behind or attached to you guys, do you see? Can you highlight condensation reaction? I guarantee you, this also applies for peptide bond formation as well as your lipid, your ester bond formation. I always want to see condensation, loss of one water molecule, then that is worth 1 mark. The second one that you always need to describe in the formation of bonds is between what groups of what subunit. So for this case, because it's glycosidic, you always must tell me an hydroxyl group of carbon 1. Do you see? I want to emphasize the specificity of which particular carbon on the subunit also matters. That is how specific bio answers must be. Between a hydroxyl group of carbon 1 of one glucosamine and a hydroxyl group of carbon 4 of another glucosamine, because that's why I say it's always 1 to 4. So for your peptide bond formation, then you will say what? OH group of a carboxyl group of an amino acid and an NH group of another amino acid. Okay, so you'll form that peptide bond. Please specify. Clear? Additional points, they say how is it formed, because this is actually cellulose. To be honest, you 1, 2, 3 will give me my mark already. But because it's cellulose, you need to further explain that any adjacent, it has a rotation. Because why? You need to show me how the hydroxyl groups, how come they can align side by side like that. You understand? Because the glucosamine needs to rotate. Clear? This is just a bonus mark. But is it still based on what we learned? Yes. I'm just trying to shed light. There will be very similar kind of questions. Not exactly cellulose or not exactly starch, but still based on what you learned. Again, based on structure and property of chitin. You know nothing about this, but they've already mentioned it's a structural polysaccharide, just like cellulose. Do you think answers will differ widely? No. That's why you just now realize that when I talk about cellulose, I say each glucose monomers rotate 180 degrees. Why? To form cross-linking via hydrogen bonding. What is the purpose for that? For bundling, to form microfibrils and eventually fibers for what? High tensile strength. Isn't it the same thing as what we are seeing right now? Another thing is that instead of your hydroxyl groups, we say polar and acetyl groups in chitin because it's in the diagram that we see there, right? So you just need to make sure, because you should know, based on your chem, OH is polar. So in this case, you just search for anything that is also polar. So in this case, it's N acetyl groups projected outwards in all directions. Isn't the phrasing the same? From interchange hydrogen bonds again. And another thing I say, the linear is also another feature, right? Do you see right now, linear is for what? So you can have bundling to form fibrils and eventually fibers. Also same thing, high tensile strength. I'm just trying to shed light. This is another molecule, but you see it's fundamentally based on what you learn. Examiners will always test you what you learn. Keywords I want you to realize, class, is another molecule, but it's also the same. Okay? Okay, but anyway, for the enzymes, right, just to let you know, mode of action, take note, mode of action, the key phrase is all down there. I feel this answer sheet is very good in describing the mode of action of a typical enzyme. So you can use this as your studying material. Okay, usually two mode of action you always talk about, the lower activation energy and either one of the hypotheses, either lock and key or induced fit, any one that is appropriate. But I want to bring your attention for 2A. So some schools never teach you about keratin. But let's say if they test you anything about keratin, are you going to be lost? No, because I'm going to show you. Okay, so the first one they have 2A. Describe one difference and two similarities with the organization of peptide chains in keratin and collagen. We already know what is, again, one difference, right? Tropocollagen, what's another name for tropocollagen, Sarah?
Triple helix.
They're made out of how many polypeptides?
3.
And then this one, what did they call this one? Called a dimer, right? So straight away made out of how many polypeptides? Dimer is 2. Okay, so do you see it's fundamentally still based on what you learn? Then the similarities will be what? Again, do you see all of them also have fibrils and fibers? And then do you see there's a staggered arrangement? By the way, you all know what staggered arrangement, that crisscross, so that they can wind very tightly with each other. So do you see the staggered arrangement for both? This dimer for keratin is not straight line chains. It's again almost like helices. Agree? So this is things that we can actually write. But I just want to bring attention. So they ask you, how is the structural organization for just part 2, A part 2, how the structural organization of keratin enables it to fulfill its function. Fulfilling its function as a what? Again, a fibrous protein. But I just want to bring your attention, even though you know nothing about keratin, you only study collagen. Just to bring your attention, collagen, what is one feature? Linear in shape, right? Agree? So that shape, right, do you agree? You always say that collagen, to have high tensile strength, must always have cross-linking. Whether is it cross-linking for your hydrogen bonding or covalent cross-links. For this one, I know nothing about keratin, but I know at least you form fibrils and fibers. So wouldn't that, again, number 1, you need to have high tensile strength. That means you must have aggregation of fibrils into fibers, which you see in the answer sheet. That's why I have high tensile strength. Isn't this aggregation of fibrils and related high tensile strength what you learn in collagen? And then another thing is, you have no OH groups. They are insoluble in water. Again, same thing. Isn't that also what we learn? Insolubility of your collagen. Or your staggered arrangement, like this glycine is the only small enough to fit into the the crowded center, right? That's why we have staggered arrangement. So we do not know what are the amino acids, but can we use terms like staggered arrangement in keratin so that you actually have that rigidity or high tensile strength? Same thing. What I'm trying to show you is that, again, examiners will always test you what you learn. Anyway, can you help me start this respective question so you can use the answers as your revision material? Start for me question 1A part 1 and part 2, 1A part 1 and part 2, and 1B. I repeat, 1A part 1 and part 2, and 1B. Please help me star 2B part 1, 2B part 1, star 2B part 1. Please also help me star, okay, question 3C part 1, 3C part 1. And then last one, please also help me star this 4C. Okay, 4C. And with that, I end off here today. There is no class next week, so you all can go back and compile what you just learned. Alright, so I just want to bring your attention to all this. Okay, now I will continue revision when I see you even in the regular class.